Hello dear students, today I am going to start with the next cell organelles, that is microbodies. In the last video we discussed thoroughly about the locomotory organs like cilia as well as flagella, and these are also helping being in some tissues for the movement of some of the materials in one direction, right? So all those things that we discussed, and let us start with the microbodies. Now these are also kind of cell organelles that are present in the cytoplasm of the cells itself, right? So what are those microbodies and how their structure is and what are the functions they perform in cells? All those things let us discuss now one by one, right? So microbodies, these are microscopic. These are microscopic single membrane bound cell organelles present in the cytoplasm of the cell, right? And these are formed from, especially, so these microbodies are formed from the vesicles of endoplasmic reticulum, especially, right? So especially the peroxisomes are one kind of microbodies which are formed like this, right? So let us see. These microbodies are present in cytoplasm and they are of different types. One is peroxisomes, gliothesis, as well as spirozomes. These are different types of what? The microbodies itself. First of all, we'll start with the peroxisomes. These are single membrane bound cell organelles formed from endoplasmic reticulum, the vesicles of endoplasmic reticulum, and these were first discovered by Rodin and Talbot are the two scientists, the one who discovered these one, and these are meant specially for the synthesis and destroy of peroxides. That is nothing but peroxide metabolism. They are meant especially for peroxide metabolism, right? So these are especially having some of the enzymes. Those especially enzymes which are present in the peroxisomes, they help in synthesis of hydrogen peroxide, right? And this hydrogen peroxide which is stored inside these one is the toxic material. So since it is a toxic material, soon after that one, there the action of another enzyme present in the peroxisome, so that act on this hydrogen peroxide to destroy it or degrade it into so the oxygen as well as water itself. Then what happens? The toxic material is removed there. There the synthesis of this hydrogen peroxide as well as the degradation of this hydrogen peroxide is being done in the same microbodies called as peroxisomes. Alright, let us write it. Some enzymes like amino acid oxidase, peroxidase are the enzymes which help in synthesis of the hydrogen peroxide over here. Hydrogen peroxide, this is nothing but hydrogen peroxide is synthesized by these enzymes. So simultaneously on this hydrogen peroxide, on storage of this hydrogen peroxide, it becomes a toxic material to the cell. Since it is a toxic material, on this one of the enzymes act, so that is catalase. Catalase is an enzyme which act on this hydrogen peroxide and convert them into oxygen as well as water. When hydrogen peroxide is degraded into oxygen and that is water, so that moment the toxic material has been destroyed. Right? So this is how peroxisomes they act upon. So they act or they help in the synthesis of hydrogen peroxide as well as the destruction of the hydrogen peroxide itself. Right? Catalase is the fastest enzyme, as if the carbonic anhydrase, right? So it is acting very fast enough. That kind of one of the marker enzyme it is present inside the peroxisome, right? And these peroxisomes also help for the oxidation of the fatty acids itself. These also help in oxidation of the fatty acids. And one more thing what do they do is they are present in the mesophyll cells, that is green cells of the mesophyll tissue. So these peroxisomes are present, and being in mesophyll cells, as you know, mesophyll cells where they are present. So if it is a leaf, then upper epidermis, lower epidermis, in between these two epidermis there is a tissue called as mesophyll tissue, right? So these mesophyll cells mainly consisting of what? Peroxisomes. So they mainly consisting of peroxisomes. What happens being in the mesophyll cells? So these peroxisomes they perform photorespiration. They help in photorespiration, or it is also called as glycolate pathway. You will study in respiration in plants thoroughly regarding this glycolate pathway, right? So this is also one more function of, this is more important aspect to be remembered here. So these are the functions of peroxisomes. And the next one is gliothesis. What are these gliothesis? These are also single membrane bound cell organelles present inside. They are also present inside the cells, especially plant cells, right? So especially they are present in plant cells. Beaver is the name of the scientist, the one who discovered these one. Beaver. He is the discoverer of these glioxisomes, and these are meant specially for glyoxylic acid cycle. Glyoxylic acid cycle they perform. According to this cycle, in this cycle what happens is the conversion of fats into carbohydrates. Fats into carbohydrates that take place over here. Conversion of fats into carbohydrates. And such kind of glioxisomes, especially they are present in fatty seeds, and they are also present in guard cells. Guard cells of what? So they are present in the guard cells of stomata, and these are also present in fatty seeds in the sense that oil containing seeds. Oil containing seeds in the sense that What are those seeds? Example for this one, groundnut is there, castor is there. In such kind of oil containing seeds they are present in, right? And these are also found in yeast cells also. Yeast cells. Gliothesis are seen over here. So there glyoxylic acid cycle that take place according to, so in this cycle what happens, the fats are converted into carbohydrates. So once again, so they become energy boosters in these gliothesis also. These are another kind of microbodies. And then now last one is spirozomes. Let us see now itself. Spirozomes are also the membrane bound cell organelles that are present as the microbodies inside the cytoplasm of the cells itself, right? Spirozomes. These are also single membrane bound cell organelles present in the cytoplasm. Of course. And these spirozomes meant especially for storage and synthesis of lipids itself. And such kind of spirozomes were first discovered by Hanstein. Hanstein is the name of the scientist, the one who discovered them for the first time, and these were named as, means Hanstein named them as microsomes. And so another scientist, Pore, named them as spirozomes. This term was coined by Pore itself, right? And these are meant especially for the storage and the synthesis of what? The fat itself. And such kind of so microbodies, they are spirozomes. Like this, these are especially the microbodies that we discussed, and later the next cell organelle itself is the master cell organelle. Very important thing. It may be asked for 5 marks itself, the explanation regarding the nucleus as well as nucleolus. So these are very interesting things. So let us study them one by one now itself. Let's start with nucleus now. So nucleus itself is the largest cell organelle present inside the cell, right? And it is the major cell organelle. It is also called as master cell organelle. Why? Because it controls all the functions of the entire cell. Especially the metabolism of the cell is controlled by this nucleus. And let us write it now. So nucleus, it is the largest cell organelle in the cell, especially in case of animal cell. In case of plant cell, usually the larger one itself is chloroplast is there, mitochondria is there. They are larger sized cell organelle. Apart from this one, the largest one is the nucleus itself, right? So whenever we consider the tonoplast membrane bound cell organelle, that is vacuole, is larger enough in case of plant cell, but it is considered as the largest one, right? So it is largest cell organelle in the cell which controls all the functions of a cell itself, right? Especially metabolism, and along with the metabolism, it controls many functions many functions like protein synthesis. So right, so such kind of functions it controls, right, through, since it contains the DNA, RNA, right, so such kind of genetical material, that is DNA, RNA, it helps in synthesis of the proteins also. So this is also one of the important functions of nucleus. Okay, so nucleus itself has different shapes over here. Shapes of the nucleus. Nucleus has different shapes. It may be oval, spherical, round shaped, right? Discoid shaped, lens shaped, right? There are different shapes that we come across over here. Oval, spherical, discoid, lens shaped, especially in the plant cells it is lens shaped, right, and little bit elongated in smooth muscles, as you have seen, little bit elongated in shape and lobed in shape in case of WBCs. So there it is lobed one. So such kind of many shapes of the nucleus that we come across here, right? And the next one, number of nucleus. Number of nucleus is also very important one. Why? Because some of the cells, majority of the cells contain only one nuclei, right? So it is uninucleate condition is there. And some of the cells that contain two nuclei, there in the protozoans. So paramecium is the best example which shows dikaryotic condition, right? And some of the cells contain innumerable number of nuclei. Why it happens? Let us see one by one what are those types of cells itself, right? First one is uninucleate. Many examples we can quote for this one. Dikaryotic condition means two nuclei are present, right, in paramecium itself, dikaryotic condition. And next one is multinucleate. Multinucleate condition is called as syncytial or syncytium condition, means many nuclei are present in those cells, right? So such kind of conditions are seen in some of the cells like skeletal cells, skeletal muscle cells, skeletal muscle fibers, in which many nucleate condition is seen over here, syncytial condition is seen over here. And so it is seen in a majority of the fungi itself. In some of the fungi, multinucleate condition is seen in case of only one cell, right? Why syncytium or syncytial condition that occurs in these cells is because one thing is, there during the cell division what happens, the nuclear division occurs, but there is no cytoplasmic division, or so that is cell division that occurs over here. See here, if it is a cell, then there is a nucleus. This nucleus divides first. So that what happens by nucleate condition that we are going to see over here. Now nuclei divided. Now there is a need of this cell division also, but it does not happen. So that what happens in this cell, there remains two nuclei. Again the nuclei divide into 4 itself, but once again the cell division doesn't occur. So what is this condition known as syncytial condition? Multinucleate condition is seen over here, right? So this is number of nuclei and position of the nucleus. Position of nucleus. In plant cells, as you know, so in plant cells what happens is usually the nucleus is pushed towards the periphery due to the presence of large vacuole itself, right? So that is position is peripheral in position, central in position. Majority of the time in eukaryotic cells especially it is present at the center of the cell itself, right? So sometimes it is pushed towards the periphery. In skeletal cells also, so there it is peripheral in nature, central in position, alright? So like this, the position of the nucleus that differs from one type of cell to another type of cells. And enucleated cells, enucleated cells, especially matured RBCs, they do not have any nucleus within themselves. And in case of in case of phloem, there sieve tubes are there. Sieve tubes do not have any what? Nucleus within it. Sieve tubes as well as especially RBCs do not have nucleus within themselves. These are said as enucleated cells. Enucleated cells. So this is all about what? So the shape of the nucleus, number of nucleus, position of the nucleus, and enucleated condition in the cells, right? So then what are the parts of the nucleus? Let us discuss now. The parts of nucleus. Whenever you see the diagram of the nucleus, there you will find the major four parts over there. One is nuclear envelope, and the second one is nucleoplasm. Nucleoplasm, or it is also called as cariolymph. Cariolymph it is also called as, right? And the third one is very important one. What is that? Chromatin network. Chromatin network. One more thing I left there is of nuclear pores. Nuclear pores is also there, right? So these are the major parts of the nucleus, and along with this one there we add some more things, that is nuclear matrix, nuclear lamina, nuclear matrix, etc. All these are the things that we study all these things. So let us see the diagram of the nucleus first, and we will study all these things thoroughly now. Let us see the structure of the nucleus and its parts itself, right? So this is only the nucleus diagram I am going to draw over here. Okay. So this is the structure of nucleus itself. This is the nucleolus, and this thread-like material what covers inside this one is chromatin network. And the fluid flood within the nucleus itself is the nucleoplasm, or it is also called as karyoplasm, or it is also known as karyolymph, right? So what are the major parts? Let us label one by one. This is nuclear envelope or nuclear membrane itself. And the second one is the fluid what is present inside this one, nucleoplasm. And this is the structure which is nucleolus. And this is the thread-like material, chromatin network, right? And here you will find the fibrous material which is attached to the inner membrane of the nucleus, and that fibrous material makes the nuclear lamina, that is nuclear matrix. It is also known as nuclear lamina. Okay, these are the major parts of the nucleus that we come across over here. So let us study one by one. First one is nuclear membrane or nuclear envelope. Nuclear envelope, right? So it is double membrane bound cell organelle, right? Nucleus itself is double membrane bound cell organelle which is made up of two unit membranes, right? It is also double membrane bound cell organelle, right? Nucleus is double membrane bound cell organelle, that is nucleus. So which contains means it is surrounded by two unit membranes. It has two unit membranes. This is one unit membrane, this is another unit membrane. The thickness of this unit membrane is about 70 angstrom units to 80 angstrom units in its thickness, right? Thickness of these membranes. So already I have told you 75 is an average one, right? This is also same as if the plasma membrane, right? So this is nuclear envelope. It is double membrane structure in case of nucleus, and it is surrounded by two unit membranes. Why it is called as unit membrane? Robertson is the name of the scientist, the one who said it as an unit membrane. Why? Because it is three laminate, right? So three laminate in the sense that two so they are made up of phospholipid bilayer along with the middle one, right? So three lamina are seen over here in this one, hence it is said to be as an unit membrane, it is called as, right? And there, whenever you observe this membrane, so there you will find what? There is a space between two membranes. There is a space between two membranes is called as perinuclear space it is. And this perinuclear space is about 200 angstrom units to 400 angstrom units in its diameter. So perinuclear space, you will find perinuclear space. There is a space between the two unit membranes. That space is perinuclear space. And one more thing you are seeing over here in the nuclear envelope, there are some pores are provided. What are these pores called as? Nuclear pores. These are called as nuclear pores. See here, nuclear pores are seen over here. So these nuclear pores are the small microscopic pores provided on the nuclear membrane. So these are about 1000 number to, so 10,000 number they are. That many number of nuclear pores are provided over here itself, right? At the position of nuclear pores, there the two unit membranes they fuse each other. See here, this is the region of nuclear pore in which both the membranes are fused each other. Right? Point to be noted. The nuclear membranes fuse each other at the site of nuclear pore. At the site of the nuclear pore, right? So this is most important fact to be remembered over here, right? And one more thing you are observing here. So this nuclear pore, so whenever you observe, so this is just like this, right? And this nuclear pore is provided with the octagonal structure. This octagonal pore, and this octagonal pore is so making the inner lining of this pore itself, and it is provided with the protein, means it is made up of a protein octagonal structure. And this is said to be as what? This octagonal structure is made up of the protein Nucleoplasmin. What it is called as? Nucleoplasmin it is called as. And so it is a cylindrical shaped structure, octagonal in structure, and this nucleoplasmin, what it forms is called as an annulus. What it is called as? Annulus. This annulus and the nuclear pore both together form nuclear pore complex. Right? See here, annulus plus nuclear pore together form nuclear pore complex itself. Very important point to be noted over here, right? So nuclear pore complex. What is annulus? It is the cylindrical shaped structure which is made up of nucleoplasmin, and that is called as annulus. So and this annulus, so along with the nuclear pore called as a complex it forms, that is called as nuclear pore complex it is called as. And this nuclear pore help in exchange of the materials like proteins, ribosomes, mRNA, tRNA, all these are the things so that are transported between the nucleoplasm and the cytoplasm. Exchange of materials like proteins, ribosomes, mRNA, tRNA, etc. So through this nuclear pore take place between nucleoplasm and cytoplasm. So this is the fact what we must remember over here. This is all about the nuclear membrane itself. What is that? Nuclear membrane is one of the important structure of the nucleus, right? One thing. And in in the nuclear membrane, the inner membrane, whenever you observe, inside the inner membrane, there you will find the proteinaceous fibrous structure that gives support, right, to the inner membrane itself. See here, already we have labeled this one nuclear lamina. See here, nuclear lamina. What is this nuclear lamina? See here, the inner membrane is supported by the fibrous material, proteinaceous material. So this inner membrane of nucleus is layered and supported by proteinaceous here supported by proteinaceous fibers that forms nuclear lamina. And these proteinaceous fiber structures which help in giving mechanical support to the inner membrane, and simultaneously it binds the chromatin network, ends of chromatin network. It binds to ends of chromatin network. It binds to chromatin network itself, right? So that is also very important feature to be remembered. And these are also, these fibers even attached to telomeres of the chromosomes. Telomeres of the chromosomes, that is the terminal part of the chromosomes, are attached over here. So it drags this chromatin material up to the inner surface of the inner membrane itself. Means keep the chromatin network so not so highly curled sometimes it makes, right? So that is the difference over here. It makes in the nucleus. Inside the nucleus, that is called as nuclear lamina, what we call it as. It itself is forming a matrix like structure, that is nuclear matrix, right? So next one, nucleoplasm. What is this nucleoplasm? Also called as karyoplasm. Karyoplasm or karyolymph, it is also called as. It is a semi-fluid. It's a fluid. So present inside the nucleus. Present inside the nucleus itself. Right? It is present inside the nucleus, and it contains many number of ingredients. It contains many number of ingredients like, so first of all, nucleotides. What do they contain? Nucleotides. It contains many number of proteins. It contains many number of enzymes. It contains even the DNA, even RNA it contains. Right? DNA it contains, RNA it contains many number of enzymes that helps for transcription. Right? What is the transcription? We'll study them later. Right? Transcription, many number of proteins it contains. Right? And many number of nucleotides it contains. Okay. So all these are the materials, along with some more materials like lipids are also present over here. Lipids are also present over here, and some more materials like phosphorus as well as calcium, magnesium, such kind of minerals are also present over here. right? Some of the minerals like calcium, magnesium, potassium, phosphorus, right? Such kind of many number of minerals are also present inside this. So fluid like structure, that is semi-fluid, that contains all these ingredients within it, that forms the liquid form of the nucleus itself, right? So this is about the nucleoplasm. So mainly it consisting of, we can add over here also, that is nuclear lamina over here. So this nuclear lamina that binds the inner membrane of this nucleus to the chromatin material, inner membrane of the nucleus to the chromosomes. So like this it helps in the binding up of these things, right? So these are all things so that are involved in nucleoplasm. And the next one is very important one, that is nucleolus, right? So after the nucleolus, the chromatin material will discuss. Now nucleolus. So this was first discovered by Fontana for the first time in 1781, and later it is named as nucleolus by another scientist. So it is named as nucleolus by another scientist, Bowman. Bowman is the name of the scientist, the one who named it as nucleolus. And whenever you observe in the diagram, this nucleolus, it doesn't have any membrane around it, hence it is membraneless cell organelle, right? It is membraneless cell organelle, right? And this is so almost round in shape, round or roughly round in shape itself, roughly round in shape, and this has three major sections. It is made up of three major parts. Mainly, what are those? One is granular structure, fibrillar structure, right? Granular structure is there, and fibrillar structure is there, and even amorphous structure is also seen over here. This granular structure mainly consisting of the proteins which forms the ribosomal subunits, right? Granular structure. Fibrillar structure mainly consisting of rRNA, especially rRNA. It is elongated fiber-like structure. It is amorphous structure. It also consisting of the proteinaceous matrix it forms, and all these regions together form the nucleolus over here. And this nucleolus, it is attached to the specific region of the chromatin network. It attaches or it is associated with one of the structures, that is NOR, N-O-R, nuclear organizer region it is called as, right? See here, so it is attached to specific region of chromatin, and that specific region of the chromatin, which is called as nuclear organizer region, that is NOR itself. It is called as NOR, nuclear organizer region, right? Especially this nuclear organizer region is seen in the chromosomes, right? at the SAT chromosomes, that is satellite having chromosomes. What are SAT chromosomes? I will discuss whenever we are studying thoroughly the chromosomes itself. See here, it is present, means this nuclear organizer region is especially present in the number 13, 14, 15 as well as 21st as well as 22nd chromosomes. It is usually seen in 13, 14, 15 and 21 and 22nd chromosome. It is usually present in, right? They are the SAT chromosomes, so there it is associated with, right? So this is the fact what you should know about this one, right? So this is about the nucleus and this, nucleolus. So this nucleolus that disappears. It disappears during cell division. During cell division, especially in the first stage of the cell division, that is, so there are 4 stages that we come across in nuclear division. So, or karyokinesis, it is also called as. And in this, in the first stage, that is, there are 4 stages that we come across, as you know. So prophase, metaphase, anaphase as well as telophase. In prophase it disappears and once again in telophase it reappears due to this nuclear organizer region itself, right? So here we write nucleolus disappears during prophase and nucleolus reappears in telophase. In telophase, this is the fact what we must remember always. Okay, so this is all about the nucleolus. Okay, one more thing regarding the nucleolus. So I left, it is very important one. So this is commonly known as ribosomal factory. What it is? It is called as ribosomal factory. Why it is called so is because it helps in synthesis of ribosomes. So ribosomes are synthesized over here and they come out of this nucleus through nuclear pores. And here the ribosomes, they help in protein synthesis by binding with the mRNA. mRNA they bind it with and they synthesize the proteins required for the cell itself, right? So that is why these are commonly known as ribosomal factories. Why? Because they help in the synthesis of ribosomes. This is most important point to be noted over here. So it is most important for NEET examination even, right? Now the chromatin network. Chromatin network. What is this chromatin network? The name itself, chroma, means colored. Chroma means colored. When it is stained, when the nucleus is stained, that moment, so this is the thread-like material which takes the stain and it becomes colored structure. Because of this reason only it is called as chromatin network, right? So this chromatin network itself is a thread-like material present inside the nucleus. Inside the nucleus, right? And it is called as chromatin network itself. And this chromatin network, so it start to condense, right? It start to condense itself during cell division. So whenever the nuclear division starts, so the division of the nucleus starts. So first of all, during cell division, nuclear division occurs, and later, so the cell division occurs. So in the nuclear division, this chromatin network start to condense itself so as to form very condensed structures called as chromosomes. Already I have mentioned in the previous classes, right? So it start to condense during cell division, especially nuclear division. It start to condense so as to form condensed structure, form very condensed thread-like structures, and those thread-like structures are called as chromosomes. Chromosomes, they are nothing but chromosomes. So it forms chromosomes. Okay. So they are called as vehicles of heredity, etc. We have seen in the last classes. So it's not a matter over here. So chromatin network we are studying here. So this chromatin network sometimes it becomes highly condensed in some regions. And this highly condensed structure of this chromatin network is called as heterochromatin. What it is called as heterochromatin, and sometimes it is loosely arranged, it is called as euchromatin. So this one is called as euchromatin. So this kind of two chromatin network that we come across over here, one is heterochromatin and euchromatin. Is the two types of chromatin that we come across over here itself. Yes, heterochromatin, it is a highly condensed, right, structure of the chromatin network, or it is so tightly condensed one. It is highly condensed, and it is loosely condensed chromatin network itself. See here, in heterochromatin that we come across two types. One is constitutive chromatin or heterochromatin, and facultative heterochromatin are the two types of heterochromatin network that we come across in constitutive. So what happens? It occurs in condensed form permanently. It occurs in highly condensed form permanently. If it is so, then it is called as constitutive chromatin network. So this is usually seen in the regions of the chromosome. You will find the region, some regions of the chromosome like a telomere. Telomere is the terminal part of the chromosome where it is highly conditioned, and centromere. Centromere. See here, this is the chromosome, what we see right here itself. And in that chromatin network is tightly coiled over here in this region. This is called as telomere, and here also it is centromere. This is centromere, this is telomere. Highly condensed regions they are called as. Okay. Facultative heterochromatin, it occurs in highly condensed form temporarily. Temporarily it becomes highly condensed form, not permanently. It is permanently, it is temporarily. right? So that kind of heterochromatin is called as facultative chromatin. Later it becomes loosely arranged, right? Later this highly condensed one later becomes what? Loosely arranged, but here it is not loosely arranged, it doesn't become loosely arranged. This is permanent one, this is temporary one. Based on that, there are two types of heterochromatin that we come across over here. See here, this is permanently not helping for any actions, any processes like. So on this thread-like material, which is made up of what? So it is made up of this condensed structure, that is, it contains what? It becomes chromatin network, becomes chromosomes, and it is containing mainly the DNA, genetical material, as well as proteins it contains. Okay, so this is the composition of this chromatin network, right? Okay. So this heterochromatin as well as euchromatin as well as heterochromatin are the two types of chromatin. So in hetero there are two types respectively. So this does not allow, so these heterochromatin doesn't allow some of the functions. What kind of functions it helps for? So it does not allow for transcription. It does not allow for transcription. So what is the transcription? Later we see. So it does not, means heterochromatin, it is highly condensed structure, it is coiled structure, there is no possibility of transcription take place over here. So here in this euchromatin, it is transcriptionally active, means this chromatin helps in transcription itself. Let us see what is the transcription actually. Transcription is nothing but the process of biosynthesis of mRNA on one of the strands of DNA. Right? So temporarily you remember this point. So this is the transcriptionally inactive form of the chromatin, and this is the transcriptionally active form of the chromatin, and this is whenever it becomes loosely, that moment it becomes, facultative, heterochromatin becomes transcriptionally active one. So these 3 points are very important points to be remembered. I hope you will certainly remember. What is the transcription? It is the biosynthesis of mRNA on one of the strands of DNA. I will show the diagrammatical representation of this transcription, temporary thing, right here itself. So this is the DNA what we have seen. This is the coiled structure. Sometimes this becomes uncoiled, means here it becomes this kind of structure. See here. This is one strand of DNA called as template. What it is called as template. This is another strand of DNA, so it is also template. So here this is DNA, actually this is DNA on which the synthesis of mRNA that takes place. See here, this is mRNA which is synthesized on this DNA, right? This is DNA and this is mRNA. This process itself is called as transcription. Transcription that takes place in the loosely arranged chromatin network, and it does not take place in heterochromatin. This point to be noted over here itself, right? So this is about the chromatin network in nucleus. Okay, and such kind of nucleus, what are the parts it contains? It contains the nuclear envelope, nucleolus, chromatin network, right, and a nucleoplasm. These are the four major structures that are present. Along with this one, nuclear lamina or nuclear matrix also we study, right? And such kind of nucleus was first discovered by Robert Brown. Robert Brown itself in 1931, right? Basically it was discovered by Leeuwenhoek first, and later, so the explanation regarding this nucleus was completely given by Robert Brown. Hence, so he became the discoverer of this nucleus ultimately. So this is all about the nucleus and nucleolus, these two cell organelles that we discussed together in this session. So, and in the next session we'll see thoroughly regarding the chromosomes. Thank you.